Johann Gregor Mendel was an Austrian friar and abbot of the St. Thomas Abbey in Brno, modern-day Czechoslovakia. Curious about how people and plants obtain certain features over others, he began experimentation to find out. Using pea plants because of their ease of specific pollination, Mendel began to observe certain traits being dominant over others. Termed Mendelian Inheritance and forming the basis of modern genetics, this is what we'll be focusing on in this presentation. To begin the process of inheriting a trait, we must start with the parent. A parent passes on their traits through gametes. If a parent has all dominant traits, the gametes will carry only dominant traits. If the parent has one dominant and one recessive trait, the gametes will have a 50-50 chance of carrying either trait. If the parent carries only recessive traits, the gametes will pass on only recessive traits. As to which gametes get paired, that is entirely up to chance. Then, using the known traits of the parents, we can determine the chances of certain traits getting paired. Using a Punnett square like the one shown here, we can then determine the probability of which traits the child will display or gain. In this case, we are showing a dominant-dominant to recessive-recessive cross, which will likely produce all dominant phenotype children with recessive genotypes. If one wants to add more traits into the mix, they can simply increase the size of the square, showing all possible variations of the new mix of genotypes. We can then use this method to determine the likely traits of a genealogy by comparing the traits of the parents on a Punnett square. In this case, we'll assume that the blue trait is dominant and the white trait is recessive. For this case, the blue parent will likely have full dominance over the white parent, with the children showing blue dominant phenotypes and having white recessive genotypes in the background. If the resulting blue dominant white recessive children breed, the likely result will be 1/4 of the children fully blue phenotypes, 2/4 being blue dominant phenotypes and white recessive genotypes, and 1/4 being fully white recessive. That means that 3 will show blue phenotypes, and 1 will likely show a white phenotype. For related but non-Mendelian genetics, we have codominance and incomplete dominance. Codominance is when two genotypes share dominance of a single organism, resulting in a halfway point between the two genotypes. Incomplete dominance is when two genotypes fight for dominance over a single organism, resulting in a mishmashed or splotchy appearance on the organism.